Hello everyone, thank you very much for the invitation. I'm very happy to be here with you. Today I will talk about the ultrasonic vocalization as an insight into animal emotions. And I will show some neurophysiology of the reward system. But first of all, I don't know if you are familiar with the ultrasounds. First of all, I'll tell you a few words about the ultrasonic vocalization. Two districts called types of ultrasonic vocalization. The first one is 22 kilohertz. It reflects negative aversive emotional state. It looks like this. Here is the frequency. Here you've got the time. So here you've got the harmonic frequency. As you can see, they are stable in the frequency. There is another part. Here is more than 2 minutes of those sounds. All right. So the second band of ultrasounds is 50 kilohertz. It reflects positive appetitive state. 50 kilohertz ultrasonic vocalization calls are considered to be a marker of appetitive state. This method is used in the studies of drug addiction, sexual behavior, and social interaction. It is well known that mesolimbic reward system is highly involved in the production of those sounds. And as you can see, they are much shorter than alarm calls. They are not stable in the frequency. Here is an example of the trill one. So this is I didn't say it. This is a fast Fourier transform. So as you can see, it's an inverted U shape. Here is the print screen of our software, RATREC software, where you can record and analyze those signals. In 2012, Wright and Clark extracted a few subtypes of those sounds, and as you can see, they got the shorts, splits, flats, downward ramp, upward ramp, complex, multi-steps, and the combination of those sounds. Here you can see that here is the 22 kilohertz, so it's quite stable. In the last experiments, we found that there are few 22 kilohertz, but all right, so let's get back to the 22 kilohertz. When the one rat is receiving electric shocks, the rat emits 22 kilohertz, and another rat can amplify those signals. It looks like this in the spectrogram. So this is the first rat. This is another rat. Rats can synchronize those signals like children in the kindergarten. Together with Ewelina Knapska laboratory, we tried to explore this subject. Actually, we did analyze the data from the Ewelina Knapska lab, and in our study, we aimed to compare ultrasonic emission in the pairs of rats between food shocks in experienced and inexperienced naive observers. Examine whether the experience of observers amplified 22 kilohertz aversive signals emitted by demonstrators, and test whether the warning sound signaling food shock changes the communication between rats. Here is, we got a pipeline, habituation, pre-exposure to the shocks, only in the experience observers, and test. We've got the warned and unwarned group it looks like this in the test. So we've got the demonstrator and the observer. In the warned group, we've got the signal. It's 1.75 kilohertz sound signaling before the food shock. So this animal, the demonstrator, got the food shock, and another one is trying to observing what is happening. All right. So what are the results of this experiment? Here are the scatter plots of the basic properties of the recorded ultrasounds. Here we've got the duration of the sounds, and here is the frequency of the sounds. So in control group, we've got only to 50 kilohertz calls. Here we've got the warned group, and here is the unwarned group, so without those signal before the foot shock. And in naive rats, we've got a lot of long 22 kilohertz. In experienced rats, we've got long 22 kilohertz, and some of the 50 kilohertz, and a little bit of fraction of the short 22 kilohertz. And in warned group, we produce somehow by adding the signal before the foot shock, the short 22 fraction. So this is the fraction. And also in naive warned group, we've got a lot of sounds over 75 kilohertz. All right, so this figure shows the fraction of the 22 kilohertz. So as you can see, we got a lot of those sounds in experience warned group. This is the fraction over 75 kilohertz. So we've got a lot of those sounds in naive warned group. And this is the mean frequency of the 50 kilohertz calls. So this 50 kilohertz calls, we got the higher frequency of the 50 kilohertz calls were demonstrated by naive warned animals. So to understand how to read this, what was happening, we have analyzed the overlaps of the signals. So we check if the signals are emitted in the same time or not. So we did the overlapping analysis, and here on the red, there are 222 kilohertz. So the fraction of the 222 kilohertz. 50 in the green one, you've got the 50 kilohertz, 250 kilohertz in the same time, and mix when you got 22 kilohertz and plus 50 kilohertz in the same time. And in the experience warned group, there are a lot of those sounds which are overlapping in the same time. So we've got 222 kilohertz, as you can remember. There are a lot of those sounds were shorter. So we believe that this is kind of sharing the distress or beginning of the panic when the animal gets some kind of hyperventilations. And in those naive warned group, there are a lot of those mixed sounds. All right, so here are the conclusion of this experiment. So the analysis of the USV communication patterns reveals that in presence of experienced observer, most of the overlapping signals were covered by 22 classes. In the case of naive warned group observer, we noticed that USV overlaps are mixed signals. So 22 plus 50 kilohertz. The sounds from the 50 kilohertz class in naive warned group mostly consist of calls over 75 kilohertz. And detailed analysis of ultrasounds showed that it's critical to divide 22 kilohertz into long and short classes. All right, so let's get back to the 50 kilohertz. Are we happy in the same way? The answer is no. There are a lot of individual differences in emission of those sounds of 50 kilohertz. There are many papers about individual differences. For example, Arens showed that there are animals that emits a lot of sounds after amphetamine injection, and there are, so they call them low callers, and there are also high callers, and there are low callers that emit less sounds after amphetamine injection. Another example is the Damgo injection to the VTA. Burdorf et al. Did the experiment where they injected Damgo. Damgo is a agonist, opioid agonist, to the VTA, and they found also the differences between animals. And separate experiments showed a strong individual differences in emission with the 50 kilohertz during anticipation food reward. All right, so it is crucial to take into account individual differences in emission of ultrasonic vocalization induced by psychoactive substances when you will design the experiment using ultrasonic vocalization recordings. 50 kilohertz emission is variable amount rates, which indicates that individual differences do exist. And this kind of gradation allows us to analyze individual differences in brain neurotransmission and creating all-to-all correlation models. Together with Miron Kursa from ICM, we did the free experiments. Actually, he analyzed, he's a mathematician, so he analyzed the whole statistics, and we've got free models that induce 50 kilohertz ultrasonic vocalization. So the first model is amphetamine-induced 50 kilohertz. The second model is context-induced 50 kilohertz. And another one is a social encounter-induced 50 kilohertz. But I'll tell you a few words about all those experiments, and then I will show you another thing about how it works. All right, so amphetamine-induced. Amphetamine is a model substance considered to have strongest properties to induce 50 kilohertz. And the aim of the study was to determine the concentration of neurotransmitters in selected structures of the rat brain after re-exposure to amphetamine administration, and find a biological basis for differences in 50 kilohertz ultrasonic vocalization emission in response to administration of the amphetamine. Yes, the experiment was very easy. So we injected first dose on the day 1 and second dose on the day 7. After day 7, the rats were decapitated. So we record the ultrasound before and after injection in the day 7 and day 1. Here are the results of the ultrasound. So this is the number of episodes. So you can see that amphetamine induces ultrasonic vocalization in the 50 kilohertz band. All right, so we have analyzed neurotransmitters like noradrenaline, dopamine metabolites, serotonin metabolites, glutamine, and GABA in a few structures related to emotional processing. It's a medial prefrontal cortex, caudate putamen, nucleus accumbens, hippocampus, amygdala, and ventral tegmental area. And here are the compounds which were significantly different between saline and amphetamine injected groups. We can see that we've got decrease GABA concentration in the amygdala, increase concentration of the metabolite of the dopamine in the CPU, and increase concentration of the noradrenaline in nucleus accumbens. It was surprising that we didn't found the increase of the dopamine in the nucleus accumbens. We did correlated distance traveled with the ultrasounds. We didn't detect any correlation between those two parameters. And here we've got the correlation between ultrasounds episodes and the neurotransmitters. And we've got the positive correlation with the noradrenaline in the nucleus accumbens and negative correlation with the dopamine metabolites in the nucleus accumbens. So we have extracted the correlation from the correlation of the levels of the noradrenaline in nucleus accumbens, and we found that these levels negatively correlates with the dopamine metabolites and dopamine in nucleus accumbens, and positively correlates with the serotonin in the nucleus accumbens and GABA in the nucleus accumbens. So we did the model of the correlation. For you, when I showed it for the biologists, they said, "Oh, what is this? How to read this?" But for you, it's super easy, probably. But I will explain in a few sentences. So the colors are the structures. So we got nucleus accumbens, amygdala, hippocampus, medial prefrontal cortex. The lines is a positive correlation. The black lines is a positive correlation, and the red lines is a negative correlation. And the width of the line is the magnitude of the correlation. And one more thing, those rounds, the green rounds is a positive correlation with the ultrasounds, and the reds are negative correlation with the ultrasounds. All right, so here are the conclusion of this of the part A. I have to check. Okay. So re-exposure to amphetamine increases 50 kilohertz. An increased concentration of noradrenaline in nucleus accumbens strongly correlated with the 50 kilohertz. We found that noradrenaline concentration negatively correlates with the dopamine and dopamine metabolite, and positively correlated with the GABA and 5-HIA, which is a serotonin metabolite in the structure. Those relationship between the emission of the 50 kilohertz USV and the neurochemical changes that occur after re-exposure to amphetamine indicates the crosstalk between noradrenaline, dopamine, serotonin, and GABA neurotransmission in the nucleus accumbens. All right, so we'll go further to the next experiment. This is the context-induced 50 kilohertz. And the aims of the study were to extract the most important neurotransmitters that are bound up with context-induced conditioning 50 kilohertz related with pharmacological reward, and find the basis of occurring individual differences in context-induced conditioning 50 kilohertz response, and of course create the model of all-to-all neurotransmitter correlation. We've got 4 groups. We've got the morphine group and saline group. The animals were decapitated before the withdrawal period or after withdrawal period. So this is the group of the with the withdrawal, morphine and saline. Why we did it? Because we found that there are a lot of sounds in day 28 after withdrawal period. And we got very less sounds before the withdrawal period. So maybe it's that context-induced 50 kilohertz ultrasounds is an illustration of incubation of craving. We analyzed those neurotransmitters, and we found that number of the ultrasounds, emitted ultrasounds in the 50 kilohertz bands positively correlates with the serotonin in the hippocampus, amygdala, and medial prefrontal cortex, and also positively correlate with the ratio of glutamine to glycine in the nucleus accumbens. So we extracted the correlation of the serotonin in amygdala, and serotonin amygdala negatively correlates with the GABA concentration in the medial prefrontal cortex, but positively correlates with the glutamate in the amygdala and glutamate in the hippocampus. What is also interesting, it positively correlates with the serotonin in the hippocampus and nucleus accumbens, and also with the serotonin in the CPU. And we extracted the concentration of the levels between the correlation of the levels of the glutamate in amygdala, and it positively correlates with the serotonin amygdala, and also the levels of the glutamate in the hippocampus positively correlates with the GABA in the hippocampus and glutamate in the VTA, and also with the amygdala serotonin in the amygdala. And serotonin in the hippocampus positively correlates with dopamine in the hippocampus, and also with the serotonin in the amygdala, yeah. All right. We also use of course, you've got the positive Spearman correlation, but also we use the machine learning methods. This is the Boruta algorithm that found some additional associations, and we can see that. The differences between 14 day and 28 days. So this is after withdrawal period. We've got increased levels of glutamate to GABA ratio in amygdala and also in the VTA. So this is the second. This is the second. Sorry, I have to check the time. All right. This is the second, the second graph. So we've got the correlation. Now you know how to read it, and we've got the positive correlation with the ultrasounds with the amygdala and also in the hippocampus and the CPU. All right. So what are the conclusions of the part B? The data collected confirms that re-exposure to the context of morphine administration after withdrawal period increases the level of 50 kHz ultrasounds. The contextual 50 kHz response is associated with the serotonin concentration in amygdala, hippocampus, and medial prefrontal cortex. The concentration of serotonin increases simultaneously in amygdala, nucleus accumbens, and hippocampus, and moreover, serotonin concentration in amygdala is bound up with glutamate level in the structure, as well as in hippocampus, and negatively correlated with GABA concentration in the medial prefrontal cortex. All-to-all analyses indicate that concentration of glutamate in the hippocampus is proportional to concentration of glutamate in the VTA and GABA concentration in the hippocampus. And presented analyses indicate that strong correlation between serotoninergic and glutamatergic system in context-induced conditioning response. And the strength of the co-transmission correlates with the number of 50 kHz emitted in response to the morphine paired context. All right, and the most central hub in this model was the glutamine in the hippocampus. Right now we are trying, because amygdala, serotonin in amygdala correlates with the ultrasounds and also correlates with the glutamine in amygdala. Right now we are running the experiments where we cut this correlation by the homogenetic methods. All right. So let's skip to the C. Let's skip to the C. So the social interaction induced 50 kHz ultrasonic vocalization. Ultrasonic vocalization are important for coordinating social behavior. In rats, an examination of those neurochemical mechanisms that govern social behavior and ultrasonic vocalization emission is crucial for understanding the social impairments occur in many neuropsychiatric disorders. To elucidate neurochemical changes in the brain structures related to social behavior and their mutual relationship, we conducted three-phase experiments. And neurochemicals were measured in following three behavioral situations: without social encounter, with short social encounter, and with long social encounter in isolated and non-isolated rats. We analyzed the same set of the neurotransmitters, and the aims of the study were to extract the most important neurotransmitters and their metabolites that are involved in social encounter-induced emission of 50 kHz, and create, of course, the model of all-to-all neurotransmitter correlation, and find the neurochemical basis of the 50 kHz USV emission during social encounter. We used the model of social isolation for 21 days, and then after the social deprivation, we've got the encounter, which induces 50 kilohertz. After this, brain were extracted and analyzed for the concentration of neurotransmitters. So the isolation induces, encounter after isolation, after social deprivation, induces 50 kilohertz ultrasounds. We found 33 significant Spearman correlation between the number of USV episodes and the compounds levels or ratio. Here are those results. I will not explain every results, but we can see that ultrasonic vocalization is bound up with the glutamate. This is we took isolated and unisolated animals together, and took the correlation of the USV. We've got the positive correlation of the glutamate in the amygdala and VTA. We've got the positive correlation with the metabolites of the dopamine in the VTA and also in the CPU after long social interaction. So the effects of social interaction of neurotransmitters levels. So what are the differences between neurotransmitters levels in isolated and non-isolated animals? So there are few differences. Depends on the duration of the social encounter, and we've got the decreased levels of the dopamine metabolites in the amygdala and in the nucleus accumbens, what was very surprising for us. And also the decreased levels of the glutamate in nucleus accumbens. When we take the animals which got the interaction, long interaction, we've got completely opposite effects. We've got the increased levels of dopamine in the VTA. And metabolites, we've got the increased levels of the serotonin in the hippocampus and the nucleus accumbens, and also increased levels in the serotonin metabolites in the CPU. The only parameter that differentiate animals which were not preceded through the social interaction was the glycine in the VTA. And the glycine in the VTA, sorry. And the glycine in the VTA rises during social interaction, and positively correlates with the dopamine metabolites in the VTA, and also in the glutamates levels in the amygdala and in the VTA. To understand what was happening, we did the stage analysis for the social encounter in all animals. We've got three time points. Yes, 3 time points. So without social interaction, short social interaction, and long social interaction, and we found that the levels of the glutamate in the hippocampus rises in these 3 points. Also, the glutamate in the amygdala and glutamate in the VTA. We've got also increased levels of the GABA in the hippocampus and dopamine in the hippocampus and in the CPU. What is also interesting, we've got the increased levels of the GABA concentration in the VTA. But to understand the neurochemical background of observed substantial increase in the number of 50 kilohertz calls during social encounter in isolated animals, because we've got the differences, all right, in isolated and non-isolated animals. All right, I have to speed a little bit. We did it for isolated and non-isolated animals separately. And we found that in isolated animals, we've got the increased levels of glutamate in hippocampus, amygdala, and glutamate. We didn't get the same. We got only increase in the glutamate in the hippocampus. All right, I will skip it because we don't have time. All right, so social interaction induced 50 kilohertz is bound up with glutamate in amygdala, VTA glycine in amygdala, hippocampus, the nucleus accumbens and the CPU, GABA in the VTA, and dopamine metabolites in the VTA and CPU. I will skip the conclusions. You've got here the neural reward to GitHub. This is the interaction table. When you can click or declick the rat group substances, structures, and effects. All right, so we've got the free models of the correlation between neurotransmitters and what happens when we put all those experiments together and find what is the background of the 50 kilohertz calls. So we've got the glutamate. So the positive effect expressed in the 50 kilohertz ultrasounds is bound up with the glutamate in the amygdala, hippocampus, CPU, and the VTA, glycine in the amygdala and the VTA, hippocampus and CPU, GABA in the hippocampus, and serotonin in the CPU and in the amygdala, and also the dopamine in the hippocampus, MPFC, and metabolites in the CPU. The most central hub in this free experiments was the amygdala glue. And here's the heat map demonstrate that social interaction is the most strongest stimulus. So the last question: is it fake or real? Do those classical statistical analysis or machine learning methods have the biological sense? So we did the first test. We used the homogenetic methods. We injected the viral construct to the amygdala and the VTA. We used the social isolation tests. The tests here is the pipeline of the experiments. So we've got habituation, surgeries, recovery, isolation, and social interaction. We found that also during this experiment that animals get the different voices by the analysis of the entropy. All right, and here are the results. So we suppress the glutamatergic neurotransmission in VTA and the amygdala, and despite the fact that the number of the ultrasonic vocalization were the same, we produced the fraction of the 22 kilohertz calls. We didn't get those calls in the saline group, and we got a lot of those calls when we suppressed the glutamatergic neurotransmission in the amygdala. Here is the spectrogram. So, and here you can see that the episode duration in this group where we suppress the glutamatergic transmission in the amygdala and VTA, we produced those 22 kilohertz aversive calls. To understand what was happening, we did the timeline. Here you've got the saline group, and here you've got those groups where we suppress the glutamatergic neurotransmission. As you can see, we've got in the beginning 50 kilohertz, and then there are a lot of 22 kilohertz calls, and we did the overlapping analysis, and we can see that there are a lot of overlaps in 22 kilohertz calls, so they amplified those aversive signals, and we've got also the mixed calls. So 22 plus 50 kilohertz. So it's a kind of same situation like in the previous experiments we've demonstrated and observed. I will not tell you more about this. We did the machine learning analysis using Boruta methods, and we analyzed those neurotransmitters, but it's totally fresh. We didn't interpret this. Maybe next time. All right, so you can see that the social life is quite complex, and we are trying to understand what is happening and what is happening during activation of the reward system. We did some step forward to understand the emotion of the animals using the ultrasonic vocalization. We know how to use it. All right, so I would like to thank Miron Kursa from Interdisciplinary Center of Mathematical and Computational Modeling, Tomasz Jaroszewski, Wiktoria Karwicka, Monika Falińska, Kasper Kukliński, Paweł Boguszewski, Rafał Czajkowski, and Ewelina Knapska. Thank you very much.
Thank you very much for this inspiring lecture. I'm very surprised dopamine did not showed up in your analysis. But yes. Before you can address that, are there any questions? We have time for one, maybe two questions from the audience.
Thank you for the interesting talk. I will take the opportunity to ask the question. So I didn't get it. I think when you inhibited glutamatergic transmission, it was both at the same time in the ventral tegmental area and amygdala, and it was mainly basal-lateral amygdala, as I saw. I think.
Yeah.
Mm-hmm. Yeah. Do you think that inhibiting them separately could have different effects?
Yes, this is a great question. As you can see, we didn't do the projection between the structures, but we tried to affect the hubs of the model. So we are planning to do separate groups with because I didn't have time to show this. We would like to do separate groups for VTA separately, amygdala separately, and combination and hippocampus and combination of those groups. Probably, I don't know.
Okay. And how the social interaction looked like?
Actually, when the animal when you suppress the glutamatergic neurotransmission in these two structures, animals don't behave in a normal way. So when one animal is emitting those 22 kilohertz, and another animal depends on this, if he's also sharing the stress, he's doing some freezing, but not the whole the time. And when another animal is emitting 50 kilohertz sounds or higher, I don't know. Maybe it's some kind of social buffering, or he would say to another rat, "Oh, be a calm guy." So it's not so scary, or something like this. Thank you.
Okay, and last question.
Thank you. Really interesting talk. I have a maybe basic question. Are the repertoire and type of those vocalizations are rats learning that in some way from other rats, or is it something that is genetically embedded in them? Is it like anything like learned thing? And whether it can be acquired from another rats that those rats are living with. Mm-hmm. So you showed that there is a high heterogeneity between rats. Mm-hmm. Could it come from the fact that they grow in different environment with different rats, and mm-hmm. could it come from the fact that they are at different level of group hierarchy.
Yeah, this is very good. This is very good questions, and we will try to we are trying to understand this. The small rats, they don't emit those two bands. They emit sounds about 40 kilohertz. Of course, probably they learn how to vocalize, but we can see in the habituation methods, because we use the tickling methods. So we tickle the animals to check if they are emitting the large number of those sounds or the less number of those sounds. The answer for your questions about if they are learning, we don't know. We didn't do the experiments in the semi-natural environments. We are planning to do this on rats, but it is very long. very long experiments. And due to the subtypes of episodes. A few days ago, we did the experiments with using the psychedelics LSD and checked what is the effects of the few doses of the LSD on the affects, and we found we just started to analyze it, but it was surprising for us that we found some ultrasounds that are starting from the 50 kilohertz, then going to this is the same episode, going to the 22, and then we've got all the 50 kilohertz. So it's some kind of I don't know how to use liquid emotions, or I don't know. We are trying to understand what was happening.
And so, for example, between different cohorts of animals, like animals that were bred in, I don't know, France, between those that were bred in Poland, you think there are different languages?
Yeah, we got a big network of the ultrasounds friendly scientists, and for example, the guy from the Italy said, "Oh, my rats don't emit trills after amphetamine." Okay, but you know, there are a lot of points that you can miss. For example, where is the microphone? Because when you catch the ultrasounds, for example, when you put here you've got the rat, here you've got the first microphone, here you've got the second microphone, and you record on these two microphones. The spectrums looks a little bit different. Here you will get, for example, trill, and here you will get the flat. From the same. So, yeah, it might be.
Thank you.
Thank you.